The title of my presentation is Expressional Alteration of Epigenetic Regulators Acting as Potential Biomarkers of Leukemia Targeted by Genistein and In Silico Approach. I am Salini, IIT PDF member. So, leukemia is one of the emerging blood cancer causes where its higher incidences and higher mortality is creating a striking danger among the global records. So there is a high incidences, means every year there's a large number of people affected by leukemia, and the death rate is also high. Now we should know that there are beyond genetics certain things that can consider whether a person will be leukemic or not. First, we should know what is leukemia. It is blood cancer. Due to increased proliferation of blood cells, the leukemia occurs. All the blood cells like WBC, the lymphocytes, and these all myeloid progeny cells are upregulated. So now we want to know that there is certain genes or chromosomal alteration that can cause leukemia. This is known. But there are beyond that certain epigenetic considerations that can cause leukemia. So what is epigenetic consideration? The DNA, which has the coded things within us, can be altered. The expression of those codings can be altered due to the methylation or acetylation markers on the DNA or on the histone, which can change the gene expression and can lead to disease condition. Now, there's a new thing, liquid biopsy. We all know biopsy is a cancer detection method. But what is liquid biopsy? Liquid biopsy is blood-based biomarker identification. Because in many cases, because cancer is a metastatic, it can circulate to the secondary site and it can cause a tumor formation. So due to that, in the blood, they can form certain circulatory tumor cells, and circulatory tumor DNA are also found in the blood. So if you can take blood from the patients, you can get the circulatory tumor DNA, mRNA, and the blood cells as well. So my objective is to identify the candidate genes responsible for leukemia, the correlation between epigenetic modulation and incidence of leukemia, and next, a phytochemical, a soybean isoflavonoid named Genistein, to be used it in therapeutic purpose. So for which my procedure was metadata analysis in the global data repository like NCBI Gene Expression Omnibus. We can get the patient records, global patient records, irrespective of the countries. We can get the records of them. And then we got the differentially expressed genes and try to understand what is the interaction between these proteins. Is there any interaction? So the interactome analysis was performed in string database, and next we wanted to use Genistein as a therapeutic purpose. So Genistein, whether it is binding with these proteins or not, for which we performed in silico docking analysis. So the entire work is a bioinformatics and AI based approach. So after going to GEO2R, it's a online database. Geo is the database where you can get all the data, and GEO2R is an online software system where R programming, a statistical programming, has been used to analyze the expressional status of different genes. So there we could find at around 250 genes were found to be differentially upregulated in comparison to the healthy individual compared to the cancerous one. So among those genes, we took the data sets of 5 in particular, where there were more than 2,000 patients. And among those patients, we identified the common genes. And we created the Venn diagram where we could find that around 13296 genes were common between 4 sets, and 3 genes were common in all the sets. So what were the 3 important 3 genes were DNMT3A, DNA methyl transferase 3A, IDH1, isocitrate dehydrogenase 1, and TET2. These 3 are epigenetic markers which can alter the expression of gene on the basis of methylation or acetylation pattern. Not only that, another important factor, EZH2 and IDH2, were also found to be differentially upregulated. Mainly, if you can see in the control, the gene expression was lesser, while in the case of cancer, as most of the patients had upregulated expression of this gene. Then we wanted to understand the interactome. Is there any protein-protein binding in between? So string database analysis, which is an interactome analyzer's browser, there we could find that there are strong cluster formation between these five. The strongest of them were between DNMT3A and EZH2 and IDH2 and IDH1. That means the multi-protein complex can combine together and can bind on the DNA to alter its topology to make it inaccessible for the further gene expression. So what are these genes doing? They can do the epigenetic regulation. That means they can alter the gene expression. They can cause the methylation in the cytosine residue of the DNA. They can cause the particularly present in T-cell lymphoma, erythrocytic leukemia, and they can cause their major integral part of the DNA methyl transferase complex that can also cause lymphoangioma. Now, because we started today with the session of phytochemicals, means plant-based chemicals, to use it to repurpose it in the therapeutic purpose. So for my work is also with plant-based phytochemicals. The soybean isoflavonoid is an extracted compound from soybean, and it's a group from isoflavonoid formula. So it helps in the anti-inflammatory, anti-cancer agent. So in other cases, in previous studies, it can prevent the prostaglandin synthesis. It can prevent the activity of nuclear kappa beta, which is a transcription factor, which is involved in the gene expression. And also it can induce nitric oxide synthesis. And ultimately it can cause differential expression of pro-inflammatory cytokines and can cause cell cycle arrest and death of cancer cells. So it can selectively act on cancer cells and can cause cell death. So by getting these prelude informations, I thought about using this Genistein, whether it can also target these epigenetic modulators in therapeutic purpose to bring about a change in the therapy of leukemia. So this is the finally we did it with AI-based tools in silico docking analysis. These are the protein structures. In protein 3D structure, all these 5 proteins were found to be highly interacting with this particular compound. You can see here highly efficient binding, and the binding scores were impressive, and we could find the ligand positioning was also very impressive. So my conclusion is that if we can get the higher identification of biomarkers, besides the chromosomal alterations and genetic expression, the epigenetic markers should be also checked to predict the risk and assess the progression of the cancer. And also Genistein, upon binding as an epigenetic modulator on the, it can become an effective therapeutic agent. So thank you very much for considering my presentation.